So hello everyone, today we're going to be talking about how you can make your own mRNA vaccine and as well as general information about plasmids.
So all of this information will be leading up to you guys making your own mRNA vaccines, but because there was a lot of technical difficulties during our in-person lesson, we decided to re-record. Okay, so first off, what is a plasmid? Now let me give you a little bit more of a backstory. So DNA is often termed the blueprint of life because it contains all the instructions an organism needs in order to grow, develop, reproduce, and survive. And now a plasmid is an extra set of instructions or DNA that often carries genes that will give the organism a selective advantage. Now, the plasmid will usually come in the form of circular piece of DNA that can replicate separate from the host chromosomal DNA and exist separately from the host chromosomal DNA. Now, an analogy that can be drawn is like with Minecraft. So in Minecraft, you can have like a plasmid would be synonymous with a helmet enchanted with fire resistance too. And when you put on that helmet, now you have fire resistance too. So you have a selective advantage over your peers, and that's similar to what a plasmid will do in another organism.
And basically, like in nature, plasmids provide one or more functional benefits to the host, such as resistance to antibiotics, degradative functions, and or like virulence, which basically means like the danger and speed of spreading the disease. And all natural plasmids contain an origin of replication, and basically this just controls the host range and the copy number of the plasmids, and it typically includes a gene that is advantage for survival. And an example is like, for instance, the antibiotic resistance gene.
So now before we move into how you can manipulate your own plasmids to make mRNA vaccines, I think you should build a little bit more intuition about how an mRNA vaccine works in the first place. Okay, let's just, nope, and it doesn't work.
Yep.
That's fine.
Coronavirus. Like many viruses, it uses a protein on its surface to attach to and enter our cells. Antibodies that fit onto this protein can block the virus from attaching. Coronavirus mRNA vaccines teach our immune system to make these antibodies. How do mRNA vaccines do that? mRNA is a genetic material that instructs our cells to produce proteins. The mRNA in the vaccine is wrapped in a layer of fat particles that protect it and help it get taken up by specialized cells of the immune system called dendritic cells. Once inside these cells, the mRNA does not enter the cell's nucleus or interact with DNA, but remains in the cytoplasm with other mRNA molecules, waiting to create the enzymes our body needs. When ribosomes read the vaccine mRNA, pieces of the viral surface protein are made. These pieces are then displayed on the surface of the dendritic cell. The dendritic cell travels to a nearby lymph node, where it presents the surface proteins to other cells of the immune system. Some of these cells, called helper T cells, train B cells how to make antibodies that will fit perfectly onto the surface protein of the virus. Other cells stimulated by the protein pieces, called cytotoxic T cells, can kill virus-infected cells. Now, when the coronavirus tries to infect us, our immune system is ready, immediately recognizing, neutralizing, and destroying it before we ever even have a chance to become sick.
Okay. Okay, so just as a summary of the main points of the video that are relevant to us. So the mRNA vaccine contains mRNA, the mRNA of which tells the cell how to code for the COVID-19 spike protein, which on its own is harmless. Now, once that mRNA vaccine gets inside of your body, specific immune cells called dendritic cells are able to take that mRNA, process it in the ribosomes, and then create the spike protein and display it on the surface of the cell membrane. So your body will recognize that that spike protein is an antigen or a foreign molecule and produce antibodies in an attempt to get rid of it. Now, the cool part is after the antigen is gone, your body will retain memory B and T cells that remember the exact recipe for making those antibodies. And the existence of those memory B and T cells is what we call immunity and is the goal of any vaccine that we have ever created. So that was like a little bit about how an mRNA vaccine works as an overview. Now into the anatomy of the vaccine itself. The slides would work. Okay, yeah. So the video did mention that it's literally just mRNA and that's enwrapped by a layer of lipid nanoparticles. The lipid nanoparticles help transport the mRNA into the dendritic cell in the first place, so they're quite important. Now you may be looking at this picture and wonder, well, where the heck did the plasmids come into play? Well, in order to produce the mRNA found in the mRNA vaccine, what scientists do is take the targeted gene sequence that codes for the COVID-19 spike protein, and they input that into a plasmid. And they allow that plasmid to reproduce a multitude of times. And as the plasmid reproduces, so does that target gene sequence. And the more targeted gene sequence we have, the more vaccine yield we will have. And the reason why scientists choose to use plasmids as a form of replication is because it provides a relatively fast, easy, and reliable way of copying that target gene sequence exactly a lot of times. Okay, so really quick, we're going to touch upon recombinant plasmids because it explains how we get that targeted gene sequence into the plasmid in the first place. It's not a natural process. We tamper with it. Okay, so a recombinant plasmid is basically plasmid with foreign DNA. So that foreign DNA is often like the targeted gene sequence that we want the plasmid to replicate. Now, in order to make a recombinant plasmid, there are 3 components involved. So we have the donor plasmid, restriction enzymes, and then DNA ligase. Quick rundown of the definitions. So the base plasmid is often a genetically engineered plasmid that is designed to make cloning easier. Now, you could use plasmids naturally found in bacteria, archaea, yeast, or some species of plants. But scientists have already taken those raw plasmids and made changes to make cloning easier. So why wouldn't you just use the genetically modified plasmids? All right. Next are restriction enzymes. So these are enzymes that are able to cut the sugar phosphate backbone of DNA. Now, every single restriction enzyme is able to recognize and identify a specific nucleotide sequence that we call the recognition site. And once the restrictive enzyme identifies that nucleotide sequence, it clamps down on it and it cuts that DNA in a very specific way. And because of the unique binding site or the unique recognition site, as well as the unique cutting pattern of every restriction enzyme, if you want to anneal 2 pieces of DNA together, you have to make sure you cut them with the same restriction enzyme, or else they would be like, the ends wouldn't match up and it'd be like jamming 2 mismatched puzzle pieces together. The final thing is DNA ligase. So this is an enzyme that is able to repair the sugar phosphate backbone of DNA. And you'll see how all 3 of these components come together in the process of making a recombinant plasmid in this video.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as ECO-R1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of ECO-R1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, ECO-R1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium.
So at this point in the presentation, you should have a general idea of how you are going to be making your own mRNA vaccine using a plasmid. And now I'm going to pass it on to Lucy so she can talk about the specific parts of a plasmid that allow plasmid replication to occur.
Yeah, so on to like the more of the specific aspects of the anatomy of a plasmid. So every plasmid has 8 main elements, which are the origin of replication, restriction sites, a promoter, terminator, inserted gene, antibiotic resistance gene, or autotropic selection marker, and start and stop sequences for translation. And now I'll just go over each of them briefly. So first up is the origin of replication, and this is essentially a DNA sequence, which allows initiation of replication within a plasmid by recruiting replication machinery proteins. And after that comes the antibiotic resistance gene, and essentially what this does is it just allows for a selection of plasmid-containing bacteria. And then the multiple cloning sites, so also abbreviated as MCS, comes into play, and this is short segments of DNA which contain like several restriction sites that allow for the easy insertion of the DNA. And generally in expression plasmids, the multiple cloning site is often downstream from the promoter. Continuing on with the like anatomy, there's just a few more. There's also the inserted gene, and this is essentially a promoter or like other DNA fragment cloned in the multiple cloning sites for further study. As well, the promoter region drives transcription of the target gene. And this is really essential for like expression vectors, because essentially what the promoter does is it determines which cells types the genes is expressed in and the amount of recombinant protein obtained. And finally, the last two are selectable marker and the primer binding site for like the parts of the plasmid. And essentially the selectable marker is the antibiotic resistant gene that allows for selection in bacteria. And something to note that's pretty important though is that many plasmids also though have selectable markers for use in other cell types. And finally, the last thing I mentioned, the primer binding site, this is essentially short single-stranded DNA, which is used as an initiation point for PCR amplification or sequencing, which is essentially like a laboratory technique used to amplify DNA sequences. And primers can be exploited for sequence verification of plasmids.
Okay, I look forward to this part of the presentation tomorrow. Oh wait, I forgot about this one.
Yeah.
Okay, this is an overview of how mRNA vaccines are produced.
The mRNA, the synthesized into a plasmid, the mRNA. The mRNA vaccine delivers this to bodies, it initiates. The good news is that mRNA are much simpler to produce in the lab than proteins. To create the synthesized mRNA, scientists insert the sequence corresponding to the mRNA
Recombinant plasmid
into a plasmid, a small DNA molecule within a cell that can replicate itself. This plasmid is put into a reactor where an enzymatic reaction triggers the synthesis of the mRNA. The synthesized mRNA is then purified and encapsulated in a lipid nanoparticle. The lipid coating enables the mRNA to reach our cells where protein production takes place, triggering the immune response. This process can be used to produce many different mRNA by just changing the sequence inserted in the DNA plasmid. Scientists have great confidence in the potential of mRNA.
Okay, that's it.
Mm-hmm.